Okay. Hi, everybody. Thanks for your patience while I let everybody kind of filter in. Um, so we're here, exam two review session. Um, we can get started. We started the recording. You should have been notified. We can go by raising your hand. It's hard for me to keep track of things in the chat because I don't know, it's just difficult, but I'll share my screen and we'll go over things. You can, of course, put things in the chat, but it's a little bit of a delay. It's better just to raise hand and I'll go through and call on people to ask questions. So who wants to be the brave soul to go first? All right, Vivian. Vivian, you're muted and I don't see you, so I'm not sure if you know that, if you're asking a question or not. Okay, Vivian, we don't hear you, so I'm going to switch to Sonia.
Hi. Okay, so my question, wait, can you hear me?
I can, yes.
Okay. My question is about the specifically tuberculosis. Um, this was on question 10, I believe, of the um essay questions
Huh?
or the long answer questions.
Huh?
So how exactly does like tuberculosis infection influence genomic variation? I was a little bit confused on that.
Oh, you know what? I'm wondering. So, how has pathogenic infection influenced genomic variation discussed with reference to TB, HIV, and plague? Okay. So, I think one of the things that's going on with that question with TB in particular is one of the lectures. Let me share my screen. Sorry, hold on. One of the lectures that I cut from the Mendelian disease genetics section this quarter because of there weren't as many classes like class periods because of veterans because of Tuesday Thursday schedule
Mhm.
um, was Tay-Sachs, and I have this one slide from tuberculosis. So, and I think I skipped this in lecture. Does that sound right? I think I had this slide and I think I said, we're not going to talk about it. I'm just going to skip over it.
Okay,
That
yeah.
ring a bell to folks in the room. So I actually think that what we need to do is for the study guide here where it says this, I think I think we should just say discuss with reference to HIV and plague. Um,
Okay.
yeah, yeah. You could also think about it in terms of Kuru, like we spoke about today too, with the PRNP gene. Um,
Okay.
but
Gotcha.
but yeah, I sorry about that. That is something that I ended up not including in lecture. So
That's okay. Yeah. Okay, I figured. Just thought
okay.
I'd
No, I'm
check.
glad you brought that up as the first question too. That's good. Um, okay, good. Should I move on to the
Yes.
next question? Okay,
Yes, thank you.
you're welcome. All right, Madiha, your turn.
Yes, thank you, Professor. I had a question about seminar readings. So, as far as like I gathered, it was Full Wiley and then the movies and then Singer. And I was wondering what you thought like our big takeaways should be from each.
Um, yeah. So, um, I think so. Let's start with bad sugar, um, which is the unnatural causes diabetes specific part of the unnatural causes series. I think that the big thing to take away from that is I would be relating that to ideas around how it basically pulls apart this idea that all type 2 diabetes is this idea of thrifty genotype, and it actually kind of highlights this idea of thrifty phenotype um, without explicitly saying that in the film, and talking about the ways that structural uh, entities, structural things that people cannot control influence the ways in which complex disease develops in their bodies. And then sort of the broad overview, the introductory half an hour episode is was just sort of couching that whole idea out even outside of diabetes. So that one was more for like understanding like broad picture kinds of things around sort of structural things that prevent that can lead to poor health outcomes in adulthood. And then the bad sugar is very specifically diabetes. And then for, and so for the Singer and the Full Wiley articles, um, sort of unlike the Gravely article, for example, that we learned in the beginning of the quarter, right? It had these like sort of three tenets. Um, I don't know if there's I, I'm trying to steer you towards like what you would want to focus on for studying, and I don't know if they're um I actually kind of mix up which Full Wiley article we read in class because I've read so much of Duena's work. Let me just pull that up because I think I often think it does the thing where it's talking about the variation in, hold on one second. Because one thing I want to point out is that when you go over the short answer essay prompts, um, there aren't any that are related to the Full Wiley or the Singer article, in particular. So, um, but Full Wiley, the big concept is going to be oh shoot, hold on.
This one was about like biopower and
It's
bio.
biopower. Thank you. That's the
Yeah.
word I'm looking for. So it's about that the big concept that you should know from that is the idea of biopower. Um, Let me just pull something up really quickly. Hold on one second.
So there's kind of not like a specific question. You just want us to tie it in where it makes sense, like that kind of thing. Like,
Well, I mean, if you look at the study guide, I don't have prompts for the essay and the short answer that relate to the Singer or the Full Wiley article. So I'm not going to ask you to, I'm not going to ask a question that specifically is only about either of those articles for the things you have to write out. Um, Rather, there you could see it on so you could see the concept of discriminant biopower on the multiple choice section, for example. Um, and so I would understand the concept of discriminant biopower and what it is. Um, and then for the Singer article,
It's the one about syndemics.
yeah, I mean, I just, I mean, you could, what I would do for that one is, I think the point, that I want you to take home with that, but again, it's not on the short answer essay prompts, um, is this idea that, I mean, you should know what a syndemic is, essentially. That's sort of the key component. So like just like with the full Wiley article, we could have a multiple choice question about what is discriminant power. Similarly, we could have a question about what is a syndemic. But you're not going to have to pull from the Singer. Like you can answer any of the short answer and essay questions without having to pull from those. If yeah.
Okay, that makes sense. Thank you.
Yeah. But you could also, sorry, I sort of stumbled over that response because I wanted to make sure I was steering you in the right way. But the other thing to think about too is like if you, like if I'm thinking about complex disease and I'm thinking about like the bad sugar documentary and then also the syndemic article, those two things and in some ways relate, right? Because the syndemic article touches upon this idea that you have social conditions and diseases that create a structure that influences not only your susceptibility to that disease, but to other diseases. And then the bad sugar documentary focuses on the social condition and then the development of disease in adulthood. So you can kind of see how the syndemic article can link up infectious diseases into that paradigm of structural biases for certain individuals in certain circumstances that influence not only complex disease but also their likelihood of contracting an infectious disease. Yeah. Okay. Vivian.
Hi, I had a couple of questions. For number 12 on the short answers, it was the three genes with mutations that affect an individual's risk for developing CDV. I know what they are, but I'm just wondering, like how much do we need to know about them? Like, for
Yeah.
example, like APOE, like do we need to know like SNPs associated with lower LDL levels or just the names?
No, you do not need to know the um
Like what they do?
what they do. No. Actually,
Okay.
let me just pull up really quickly. Do, do,
I
do. Let me just
think
I have
those are
it.
the three. Yeah.
Like, let me just because it's just for people's reference too that might have also been wondering about this. Heart disease.
Yeah, and then also same with the three genes with mutations that can cause FH.
Yeah. So if it's this slide, three genes with mutations that cause familial hypercholesterolemia. Know that there are three. We focus specifically on the LDLR. So that's the one where we actually learned how that is modified such that it impacts LDL uptake into the cell and out of the bloodstream. Um, you do not need to know which mutations they are because then we went into detail. Like, so for example, I actually want to show, yeah, so this pie chart, for example, like you don't need to know these. But my point is like for this, um, this is a little bit different because this is we call it the French Canadian deletion. Um, so I think knowing this slide is good, but it kind of gets at this idea of founder effect, which is
Mhm.
kind of key to the course concepts more generally. For this, it's important to know that we have population-specific variation that leads to
Mhm.
FH. And then we see that in Lebanon, we see that in Finnish patients as well. And then for where we started, which is the ACE2, let's get the others. Oh no, and then we went into, oh no, now we're in Finland. Sorry. This slide.
Mhm.
So for this one, it's the thing I want you to know about cardiovascular disease is that it is complex in the sense that multiple mutations in multiple genes. So it has allelic as well as genetic heterogeneity. And these are the three that I mentioned.
Mhm.
So if you looked at a list and there were four choices and it was like, which one of these genes has not been associated with higher risk for cardiovascular disease? And it was like ApoE, ApoA5, ACE, and then CFTR, you'd be like, yeah, cystic fibrosis gene has not been associated with increased risk of cardiovascular disease. That's the kind
Sounds
of
good.
level I want you to know this on.
Oh, sweet. And then I also had a question about the key terms that you listed.
Uh-huh.
I can put it in the chat. This was
Sure.
what was listed on the thing. We learned about G6PD, but I don't know if we learned like all the specific stuff
We
about
didn't.
that. Okay.
So for that one, um, here. Actually, no, it's in thalassemias. Yeah, so all I lectured on was this, sorry, let me close the chat, was
Yeah.
this slide. So
Okay.
the thing that, so the, so in the context of malaria, we have sickle cell anemia and mutations to the beta globin gene. We have alpha thalassemia and beta thalassemia with mutations to alpha thalassemia and beta thalassemia, alpha globin and beta globin, excuse me. Then we have evidence from G6PD that malaria has impacted the evolution of our genome. And then we have evidence from the Duffy locus. So all of those together kind of build this evolutionary narrative that our genomic material has and our biology has responded and evolved in response to malaria as a selective pressure. Um, but G6PD deficiency, you should know that it protects against severe falciparum and that you should, and that it shows evidence of selection in response to that.
Sounds
Yeah,
good.
but don't
And
worry about
then
Fauvism and all that stuff in the chat there.
and then one more question for alpha thalassemia. We don't need to know like the different like AAs, wild type A, like yeah, all that stuff.
No, I mean, I
Just
think the
confirming.
big thing that you want to know about alpha thalassemia, right, is I'm in the beta thalassemia zone. I'm in
You're good.
alpha thalassemia. Yeah, I mean, you want to know that like ah this is important that you have
Mhm.
four alpha globin genes. And because you have four alpha globin genes, you have this variation in how severe your alpha thalassemia can be.
Mhm.
Um, but you don't need to know these details that are here.
Sweet. And
Yeah.
then adding on to that, like question 11 asks like what are the two types of thalassemia, blah blah blah. How do these changes impact the structure of hemoglobin? Is like kind of just a general thing, like the mutations lead to like imbalance globin chain production because it's like for alpha, then you have less alpha globin, and then you have like an increased amount of beta, then it's uneven. And like a normal hemoglobin has the heterotetramer, blah blah, blah,
Yeah, that's
right? Or
exactly. Yep. Yep. So it has two beta globins and two alpha globin chains that make up that
okay. So when
heterotrimer.
you have thalassemia of either one, it just leads to imbalance globin chain production doesn't really work, causes anemia.
Yes. And
Okay.
the severity of that depends on how many
Hmm.
things you're missing.
Hmm. Yeah.
Um,
Okay.
and also know too, I think it's important, right, that if you have four non-functional alpha globin genes, it's like neonatal death. Like it's non,
Okay.
um, yeah.
Thank you so much.
Yeah, you're welcome. I'm gonna stop my share just because I'm gonna go into the chat because I saw, hi, for the sickle cell treatment, it would be chemotherapy, gene therapy, and bone marrow transplant. Um, I think that's right. I just want to make sure we're not missing something that I'm not thinking of. I mean, I think that the big thing that I emphasized this year was gene therapy. But yeah, those are the only three that we discussed in lecture. Yeah, that's correct. Is the cause of sickle cell anemia a mutation in the HBB beta globin gene or a missense mutation at Okay, so sickle cell anemia is caused by a missense mutation at amino acid position 6 of the HBB gene. So it's all of those things. Could you explain question 10 about how, for example, for TB, should we talk about heterozygote? Okay, Jessica, I think you joined after we actually had a question on this, which is no problem. There is a slide on tuberculosis and Tay-Sachs, but I actually skipped over that in lecture. So I would just modify question 10 to say HIV and plague, because we didn't really talk about tuberculosis in the context of genomic variation. Um, yeah, yeah. And you could add Kuru to that list because we talk about how Kuru influenced PRNP variation. Um, so yeah, please, Mariha.
I know we did our breast cancer lab during section, but for number 8, can we kind of touch on which risk factors you want us to focus on? Because there's like quite a few for
Yeah,
females.
okay, yeah. So the big ones, am I still sharing my screen?
Nope.
Yeah, I'm just gonna show you. Share. Yeah, so number 8 is mutations in what genes increase risk of breast cancer. So that's your BRCA1 and BRCA2. But then there's a bunch of other ones too, right? But those are the BRCA1, BRCA2. What are the major environmental risk factors? So contraception, age, exercise, BRCA1, BRCA2 mutation, I think are the big four from that lab that you should know. Yeah. Um, Aviana?
Hi, I had a question about it was on the vocab concept list under
Uh-huh.
thalassemia.
Uh-huh.
Can you explain the unequal crossing over concept?
Oh, yes, I can. And I'm going to do that with a screen share. Do, do, do. All right. I just, so I kind of feel like I kind of feel like I don't have that slide. I think you can just cross that off.
Okay. I also had one more question under thalassemia. Uh, how much do you want us to know, like, about inclusion bodies?
Oh, what I want you to know is that you can visibly see. So here sorry, I don't want to show that image. I want to go here, here. Um, I just want you to know that beta thalassemia and alpha thalassemia, when you look at red blood cells under a microscope, they actually look different.
Okay.
Yeah,
Okay. Thank
yeah,
you.
yeah. So question is, how is the HBS allele an example of balancing selection? And can we go over number 10 for HIV and plague? Yes. Okay, so how is the HBS allele an example of balancing selection? So the HBS allele is, I want to show you, wait, where's the slide that I don't understand why those slides are just like, okay, here we go. Okay, so if you are a heterozygote, you are slightly anemic. You have some sickled, half of your cells can become sickled, but the other half are not. But all those cells that are sickled, if malaria gets in those cells, it can't break down the hemoglobin because it's in those inflexible chains. And so malaria can't reproduce in those cells. It can only reproduce in the cells that are the non-sickled. So basically you have like half the amount of malaria that somebody who was homozygous for the HBA allele, which is the absence of sickle cell. And then if you are HBS, two HBS alleles, and you have all of your cells sickled, right, you have sickle cell anemia. So balancing selection is where both the allele for the rhombus red blood cells and the allele that encodes sickle cells are both present in the population, and they're both maintained because the phenotype that has the highest fitness in an environment with falciparum malaria is that heterozygote that's 50% sickled and 50% not sickled. Does that get at your question? Okay. And then can we go over number 10 for HIV and plague? Yeah, so it's CCR5 delta 32. Is CCR is HIV and plague. So so for this one, we talk about the chemokine in the plague lecture. We go in detail over what the CCR5 chemokine receptor 5 is, right? It's one of these transmembrane proteins that is expressed in T cells and macrophages. And there's a deletion that's 32 base pairs long that makes the CCR5 protein non-functional. So your macrophages and T cells don't have this protein that is on the extracellular surface. And we see that it's at really high frequency in Europe. And when we look at this slide, so this is looking at data that is modern people alive today who live in Germany, who are Germans. And then we have archaeological samples, historic Germany, Sicily, people who died of the plague, people who died just before the plague, in the Great Famine, and then we have individuals from the Bronze Age. And so these data suggest that the frequency of the delta 32 allele was already high prior to plague. But I have this little note down here. What we are now understanding is that Yersinia pestis didn't first show up on the infectious disease scene with the Black Death. That there were probably previous plagues, previous infections with Yersinia pestis, even though it wasn't on the level of the Black Death with the amount of epidemic proportion that Europe experienced in the 1300s. So plague didn't drive the frequency of CCR5 higher necessarily. Sorry, let me rephrase that. The Black Death did not drive the frequency of the CCR5 allele up in particular, but that doesn't eliminate the possibility that the bacteria Yersinia pestis did not influence CCR5 delta 32 mutation frequency prior to the Black Death. And then with respect to HIV, the wording of that question is a little bit weird for HIV, because we don't have, I didn't talk at all about examples of mutations that have increased in frequency because of exposure to HIV. That's not something that we know or that we discussed in class, because we don't, there's no example, there are no examples of that. What we do know is that going back to CCR5 delta 32 is that CCR5, if it's missing, you are protected from infection with HIV because HIV can't enter the macrophage. So it offers a protective effect for individuals who don't have that protein because they have the delta 32 mutation. Okay. Judy asks, would you be able to explain post-translational histone modification, DNA packaging, acetylated versus deacetylated? Yes, so let's go over epigenetics. And for that, we need to go to the diabetes lecture. So everything on that topic is in the second half of the diabetes lecture. And we talked about two mechanisms of epigenetic modification. So that's stuff that sits on top of the genome as opposed to changes to the actual base pairs. And DNA methylation and histone modification are the two that we talked about. Mostly we spend our time talking about DNA methylation. We know, like we collectively as humans, as scientists, we know much more about DNA methylation than we do any other epigenetic process. We've studied it a lot more, like we have a lot more data on it. And DNA methylation is when this methyl group is added. You can see it here. This hydrogen ends up with two more plus a carbon, and that's what's called a methyl group. So you go from a cytosine to a methylated cytosine. And when that happens, if it's a methylated cytosine like shown here in this image, it usually leads to closed transcriptional access to that particular part of the genome. So the expression of that gene is curtailed in comparison to regions of the genome that are not methylated or unmethylated. And the machinery that needs to have access to the actual base pairs can have access, and those genes can be transcribed. And so if you have more methyl, the idea is that the gene has low expression, or you sort of turn off that protein. And if you take away methyl groups, or you have less methyl, then you have open access for the transcription machinery, and you have more expression of that gene. With acetylation, it's actually the opposite. So you can have histones, which are the structures that the DNA is wrapped around, and those histones can be deacetylated or acetylated. And when they're deacetylated, they're actually really densely packed, and when they're acetylated, they're loose. And so the same concept applies. The transcriptional machinery can access the genomic material when you have more acetylation, because it's kind of open, and so you have more expression of the protein product. And when something is deacetylated, it's more densely packed. So it's difficult for the transcriptional machinery to access, and the enzymes to access the DNA content to express the gene. So more methyl, less expression, more acetylation. More methyl, how do I want to say it? I want to say more methyl, less expression, more acetylation, more expression. So they're flip-flopped. Okay. All right, I got a thumbs up. Sweet. Thank you. That really helps. Okay. Oh, you know what? You can, so yeah, so case control association study and genome-wide scan regarding heart disease, just cross that off the study guide. Um, and you can also, we didn't actually go into, I don't think I got a chance in lecture to talk about the blue zones, which were Japan, Italy, Greece is on there. I don't think I have that on the study guide. Loma Linda. Just, you don't have to worry about those either. I don't mean to say green zones, they're called blue zones. Um, so just you can those are places where people basically like have community or in faith and or faith community, eat well and exercise, and then they live really long lives. uh, yeah. Rachel. Okay, Rachel's gone. Maya.
Hello. I'm sorry if you have answered this question because I came in late.
No problem.
For the number 9 on short question, it says for each of the infectious disease that we have discussed in the course, does it mean we count all the infectious disease from the beginning of the class or only the infectious disease after the first midterm?
Yeah. I mean, for I think you can answer that question with just using materials since the first midterm. Um, you certainly wouldn't be docked points if you talked about something we talked about in the class but before, but you also wouldn't be docked points for not doing that. So the ones we've talked about in the second half of the course are malaria, plague, HIV, and tuberculosis, and Kuru. So there's actually 5 because malaria, even though we talked about it in Mendelian disease, is an infectious disease we learned a lot about. Does that help?
Okay, thank you so much.
Yeah.
Yes.
Good. Okay. Rachel asks, okay, you got a lot in there, Rachel. Can you explain malaria treatments? All right. I don't think we've even talked about malaria treatments. Did we talk about malaria treatments? I don't even think we really talked about malaria treatments. Um, so just don't worry about that. Can you explain how to answer this question? What is a prion? Okay, so Rachel, your questions in the chat are basically everything that I went over in lecture today. Um, so um, I don't know if I if I just want to like I don't know if I want to totally just like re-give everything that I maybe if you could reformulate maybe like a specific question you have about the course content. I'm happy to go over that. But uh that the question you have in the chat is like so long and big that I'm not sure really where to start. So I'll let you think about it and we'll come back to it. Um, any other questions? Yes. Okay. Got one in the chat. And then I see your hand. I'll I'll do that. I'll do you. Okay. So for Lauren asks, for hemoglobinopathies, what level of detail should we know? Do we need to memorize exact gene combinations? So you need to know sickle cell is a mutation to the beta globin gene, amino acid position 6. Sometimes I think it's helpful for the visual people out there to okay, so for sickle cell, this slide is your friend for this question. So amino acid position 6, a glutamic acid switches to a valine in the beta globin gene, causes one of the hemoglobinopathies, which is sickle cell anemia. Then for the other type of hemoglobinopathy, right, we have alpha and beta thalassemia. And you should understand that essentially this slide for beta thalassemia is you have a deficiency of beta globin chains. So you end up with a bunch of alpha globin. You have like your tetramers are all alphas. And then it's the opposite for alpha thalassemia. You have it's this slide. You have a mutation in your alpha globin gene, so then you have tetramers of hemoglobin that are all beta globin. And the other important thing to know about the thalassemias for alpha thalassemia in particular is that you have four alpha globin genes in your genome, two from egg, two from sperm. And so it creates this range of variation in severity of alpha thalassemia, ranging from asymptomatic carrier to essentially non-viable neonatal or immediately postnatal death. So I think that covers it. And then okay, and then Judy asks, and then actually Madiha, why don't you go and then I'll come back to the chat because I'm getting more in here and then I'll put my attention there.
Yeah, I just had a question for number 8 for the short answer about breast cancer, and it's saying when comparing environmental and genetic risk factors, like which of these has a greater effect?
Yeah, so it's kind of, I'm glad you asked that question because it's not so easy, right? If you have a BRCA1 or BRCA2 mutation, you are at much higher risk of developing breast or ovarian cancer than somebody who doesn't have that mutation. But only like 5% of all breast cancer cases are caused by mutations in BRCA1 or BRCA2. The majority of breast cancer is not caused by mutations in BRCA1 or BRCA2. So in those instances, right, it's environmental exposure. And there's different, essentially, there are multiple different environmental exposures that a person could be exposed to in their lifetime that could influence their risk of developing breast cancer. And the ones that I want you to know about from the lab are, the other one, sorry, I did forget one, age at menarche, overall age of the individual, whether or not you are on or taking oral contraceptives, and then exercise, and then BRCA1, BRCA2 mutation. Yeah. Okay, so now I'm going to go to the chat. Could you explain the difference between thrifty phenotype and thrifty epigenotype? You know what? They're kind of the same thing. So there's not actually really a difference. It was just an updated modification of the thrifty phenotype. So when I go to the diabetes lecture and we go down here. So the thrifty phenotype hypothesis was put forth in the 1990s, essentially, before we really were looking at epigenetic markers. Like we didn't really have a mechanism to study them at that point, like with like laboratory assays. And so the thrifty phenotype hypothesis, poor fetal, early postnatal nutrition, set in motion this idea for nutritional thrift. The mechanism through which the developing fetus potentially biologically is responding to that poor nutritional environment is very likely through epigenetic changes to their genome. So we can actually add something to the thrifty phenotype hypothesis by saying it's probably thrifty epiphenotype, because it's very likely the functional mechanism for the thrifty phenotype is epigenetic changes that take place during fetal development in response to poor nutrition, no nutritional intake. So they're not really different. It's just like, it's like a continuation and a refinement of how the thrifty phenotype could actually be borne out biologically in a body. And then how does, oh, I need to go back to Judy's question. Sorry. Is it's going, so this exam is exactly, so the question is, what's the structure of the exam? It's the same as the last one, exactly the same. So 3 short answers, 1 essay question, but you get to choose which essay you want to respond to. You get 2 choices. And then the multiple choice set. So, and then, yeah, short answers, 2 to 3 sentences. Essays, you say about 6 sentences. I don't think that's true. I think the essays are longer than 6 sentences. I think they're more like, I don't know. I don't think about them in sentences, but like, I think they're kind of like, they fill up like a page and a half. So I would put it that way, as opposed to in sentences. And then the next question from Diana is, how does the genotype phenotype have a selective advantage to malaria regarding the HBB gene? Does this have to do with sickle cell anemia and how? Yeah, okay, so the reason why you have a selective advantage against malaria is because when you have sickled cells, what's happening is that your hemoglobin are turning into these inflexible hemoglobin chains, and the malaria parasite needs to consume hemoglobin in order to reproduce, and it can't consume those inflexible hemoglobin chains. So when you have sickled cells, you have inflexible hemoglobin chains. Malaria can't consume those chains. It halts the malarial life cycle at the blood cell stage. Explain how Duffy genes related to malaria. Yeah, and for that, I'm just going to go back to, sorry. Ooh, let me stop my share. This is Hazem. Hazem asks, could you explain how Duffy genes relate to malaria? So let me go to, sorry, wrong lecture. Okay, the Duffy receptor is this, okay, so we learned about CCR5 and how, I'm going to diverge for a second and then I'm going to come back. We learned how CCR5 is expressed on macrophages, and if you don't have that receptor, HIV can't bind. So if you're missing that protein, you don't have that thing on the outside of your macrophage. Duffy is the same situation. So if you have a mutation in your FY gene, which is short, it's basically Duffy, in your Duffy gene, you don't express the Duffy protein on the surface of your red blood cell. And Vivax malaria has to dock to that Duffy protein in order to enter the red blood cell. So if you have a if you are Duffy null, which means you do not have the Duffy protein because you have a mutation to the FY gene. Actually, sorry, it's the Duffy antigen receptor for chemokine. It's the DARK gene. You have a mutation in DARK. You do not express that protein. 5X can't get into your cell and it can't reproduce, and therefore you have essentially protection against Vivax malaria. And when you look at the distribution of the Duffy null allele, you see that it's at really high frequency in malarious parts of sub-Saharan Africa. And as you move both north and south from that region of high malarial endemicity, you see a clinal distribution of the Duffy null allele. It's in fact almost fixed in, like, it's 95 to 100%. So like people like almost 100% of the people who live in this region of the world do not have their DARK protein whatsoever. Like, it's just not there because malaria, it's like it evolved to evade malaria. Vivian, I see your hand. I'm going to take your question now before I go back to the chat. Thanks for being.
Hi, of course. It's the same. I put the question in chat too, but
Okay.
it followed up with a question someone asked earlier about question 6. I know you talked about how you said the Black Death or like the plague doesn't necessarily increase, didn't necessarily drive the increase of CCR5. But I was just wondering how you wanted us to answer the question. Then it says what past diseases may have been selected
You can
for
say, you can
CCR.
say, you can say plague.
Okay,
Yeah,
so
yeah,
we
yeah.
should.
But my distinction is it's not the Black Death,
Okay, but it's the
right? But
plague that.
Yersinia pestis, as a
Yes,
bacteria, could have driven because
okay.
they may have selected for the variant. Yes.
Okay, sounds good. Thank you.
Yeah, you're welcome. Sorry, it's so much easier for me to see my chat when I'm not in the share. So sorry for the back and forth. Okay. Judy, on the vocab and concept list, you listed the the transition to active TB and the cycle of TB transition. Would it be almost the same answers since both would mention symptoms and such or no? Oh, yeah, I see what your question is asking. Like, I have 2 points. I have 2 bullet points on the study guide. One is transition to active TB, and one is the cycle of TB transmission. Are you asking if that's really kind of the same thing? Yeah, it kind of is the same thing. I want you to know. So for tuberculosis, for latent, you should know that there's latent and active TB. And sorry. Um, and I'm thinking really about too far. Thinking about this slide. Right, where you are exposed, your body basically can kind of like curtail that infection through its immunological response. And basically, it can like wall off the tuberculosis bacteria in your lungs. And so that it can't really reproduce, it basically goes dormant. That's latent TB. And then it can be activated in your lifetime, or maybe it never will be. But you can also and then it turns into active TB, and that's when the tuberculosis bacillus starts to reproduce and spreads throughout your lungs and then out possibly to other parts of your body. You could also be infected on initial exposure, and maybe your body is not able to like cordon off those tuberculosis bacteria into those granulomas, and you just have active TB from the get-go. So no latent and active, know this. And the other thing, right, is you should know that latent TB can and should be treated. It's a lot easier to treat than active TB infection. Um, okay. We have 4 minutes left. So we definitely have time for one more question if there's one out there. All right. I'm gonna sit just another minute.
Hello.
Hi, Maya. Yeah, please
Yeah,
go.
this one is just like a confusion. So for the short question number one, how does the sickle cell anemia protect against infection from Plasmodium? So on the slides, there is one slide that how it protects. So do you want us to answer like how it infects the red blood cells, use the amino acid, degrade it, but then in sickle cell it cannot degrade it, that's why it protects against? Would it be the like enough for that question, or do you want us to also include how the HBES and HBS or like
Oh, so
I'm
for
just
that
wondering.
question, I think if you're preparing for the exam and you're looking at question one, I would just
Mhm.
prepare my response to talk about how it can't degrade the amino acids, it can't degrade hemoglobin, and then you should know HbA, HbS, and HbAs. Like you should know
Mhm.
those three things, and be able to talk about them. Yeah,
Okay, then
yeah,
yes.
I would say that. And
Mhm.
then, yeah, Mandy, how you have your hand up?
Yeah, I had a question like what should our focus be for how agricultural innovations have impacted the spread of malaria? Like, is there specific
Yeah,
example?
yam agriculture.
Yeah, yes, okay.
Yep.
Awesome.
All those slides on yam agriculture. Yep. So, irrigation with big troughs, increased breeding grounds for mosquitoes, clear cutting forests, and then also the interplay of yam taboos because yam contains thiocyanate and that actually helps. I don't know why actually, but that chemical compound actually lessens the effect of the sickling. Like you have fewer sickled cells, right? Because cells become sickled over time. They don't start out that way. And
Mhm.
so there's yam taboos during during the malaria season, so that you basically rely on your cells to be sickled to help you not get malaria. So know that concept, that whole like yam, thiocyanate, agriculture, whole story. Yep.
And
Uh,
then,
yeah,
sorry, for the number 12 on cardiovascular disease, and it says like what three genes that harbor mutations. So like we should be focused on APOE and like
you should
APO.
know for cardiovascular disease. I so I'm just gonna really quickly share my screen because there's one slide in particular. Um, it's this one. ApoE, ApoA5, and ACE. Like those are the three that I mentioned. You don't need to know what they do. Like you don't need to know that ACE controls blood pressure, but I want you to know that genetic variants in these genes have been associated with increased risk or decreased risk of cardiovascular disease.
Okay, and then is that the same as because there's two questions on like the three genes that
Then there's
harbor.
a different set of genes for familial hypercholesterolemia.
Okay.
Um, yeah, and that is, sorry, it's before and I'm going the wrong way. That's this one. But the one you really need to know about is LDLR, because that's one that we talked about um, with with how LDL can't bind, and it can't. And sorry, yeah, I'm saying that correctly. LDL can't bind in the extracellular space to get into the cell where it needs to be used for uh, um, uh, plasma membrane synthesis, which is what we need cholesterol for.
Okay, awesome. Thank you so much.
Yeah, you're very
Just
welcome.
a quick question to add on.
Yeah,
So if we're answering the name of those genes, that APOE, APOA5, let's say if it is on short question,
it's not going to be on
uh,
short question. Well, oh, I
uh,
see why you're saying that because
uh,
well, okay, let me take that back. Um, yeah. Okay, keep going, keep going. If it's on short
uh,
question,
if it's on short question, do we need to stay explain like on the side of APOE, there's like cyclates on low density lipo protein and stuff. So if it's on like the assay, I'll definitely put the little sites definitions for all the genes. But if it is on short, can we leave that out?
yeah, I mean, I, I sorry, I feel like that's a very, very specific question, and I guess I'm not quite sure how I want to answer it. So, I mean, um, I think what I want to say about that question is that I'm, for question 12, I'm asking you, what are the three genes that harbor mutations? I'm just asking you to name those genes. So the expectation is not that you know all the details about what those genes do. And the reason why I'm asking this question in the short answer slash long essay question portion is that what the point is that it's highlighting that it's multiple genes that can have mutations that lead to cardiovascular disease, right? It's not a Mendelian disease. There's multiple genes that can influence your risk for CVD. So I want you to know there's three of them I mentioned. So know those three names. Um,
Okay, got it. Know the
yeah,
three names.
yeah. Know those three names. Um, and then be able to define what familial hypercholesterolemia is, and then It says, what are 3 genes that harbor mutations that cause FH? So you can talk about then, so you can say LDLR, but that's the one that we really we learned how the LDL receptor has a mutation. We went into detail on that one. So that one probably I would elevate above the other 2. Yeah.
Got it. Thank you.
Yeah, you're welcome. Okay, guys. Thank you. You're going to do great. I hope this was helpful. And I will see you guys on Thursday. Bring a pencil. That's all you need. I'll have extra paper. Okay.
Are you gonna modify the study guide sheet? You said you're gonna modify later.
When did I say that in the review?
Modify is like I don't know. Didn't you say that? Because I came in like the half of the Zoom and then
No,
I
I
heard.
don't think I said I'm going to modify it. No,
Okay.
I'm not going to modify it.
Okay.
Everything's there. Yeah, thanks for double checking. Yep, it's gonna stay the
Okay.
same.
And then you're gonna upload the video, right? This Zoom
Oh,
video.
yeah. I'll upload the video. Yep.
Okay.
Yep.
Okay. I'll make sure to rewatch it. Thank you.
Okay. Take care, everybody. Good night.
Thank you.